The first major step for the DNA replication to take place is the breaking of hydrogen bonds between bases of two antiparallel strands. The unwinding of the two strands is at the starting point. The splitting happens in places of the chains which are rich in adenine, thymine, and cytosine and guanine. Helicase is the enzyme that splits these two strands. The initiation point is where the splitting starts, which is called the origin of replication. The structure that is created is known as the replication fork. I didn't do that right.
One of the most important steps of DNA replication is the binding of RNA primase in the initiation point of the 3'5 parent chain. RNA primase can attract RNA nucleotides which bind to the DNA nucleotides of the 3'5 strand due to the hydrogen bonds between the bases. RNA nucleotides are the primers for the beginning of DNA nucleotides.
The elongation process is different for the 5' to 3' strand and the 3' to 5' strand. The 5' to 3' template is called the leading strand because DNA polymerase can read the template and continuously add nucleotides.
The 3'5 template cannot be read by DNA polymerase A. The replication of this template is complicated and the new strand is called the lagging strand. In the lagging strand, the RNA primase adds more RNA primers. DNA polymerase A reads the template and lengthens the bursts. The gap between two RNA primers is called an Okazaki fragment.
In the lagging strand, the gaps are closed with the action of DNA polymerase adding complementary nucleotides to the gaps and DNA ligase adding phosphate in the remaining gaps of the phosphate sugar background.
The last step of DNA replication is the termination. This process happens when the DNA polymerase reaches the end of the strands. We can easily understand that in the last section of the lagging strand, when the RNA primer is removed, it is not possible for the DNA polymerase to seal the gap because there is no primer. So the end of the parental strand where the last primer binds isn't replicated. These ends of linear DNA consist of non-coding DNA that contains repeat sequences and are called telomeres. As a result, a part of the telomere is removed in every cycle of DNA replication.